So guys, so far you understand the mitochondrium, you understand the ribosomes, and now we are doing the rough endoplasmic reticulum. Just want this lesson. So after this session, don't come back. And okay, before we can even be specific to say we have the rough endoplasmic reticulum, I just want us to talk about the endoplasmic reticulum because we have two types of it. We have the rough endoplasmic reticulum and then we have the smooth endoplasmic reticulum. And I want you to listen to me clearly. Don't confuse the two. They are not the same. The only reason why this one is rough is because it has ribosomes attached to it. And the only reason why this one is smooth is because it doesn't have ribosomes attached to it, right? And then what you have to know is that your endoplasmic reticulum is just it's like a tube. Like you can see these things, it's like tubes. Like your endoplasmic reticulum is just a fluid-filled channel or fluid-filled tube, right? It is continuous with the nuclear envelope. Like this is okay. Let me use different color. This in blue here, I don't know if you can see it. That's your nuclear membrane. So your endoplasmic reticulum, to be specific, the rough endoplasmic reticulum is continuous with the nuclear membrane. So it continues from the nuclear membrane, right? And then what you have to know also about the endoplasmic reticulum is that it serves as a circulatory system for the cell, right? So what's the major role of the circulatory system? It moves, it transportation. So the endoplasmic reticulum serves as a circulatory system for the cell. They will ask you that question. Which organelle serves as the circulatory system of the cell? The answer should be endoplasmic reticulum. You don't have to say rough or smooth, just say endoplasmic reticulum. Are we clear? The endoplasmic reticulum serves as a circulatory system of the cell, meaning it transports proteins. Are you now clear, Lundi? Okay, thank you. Guys, others didn't come back. I don't know why. Maybe they're tired. You see, this is what I don't like about recording sessions, because others take advantage. They don't attend and then they will go to, maybe if I upload them on YouTube, they will go to YouTube at their own time, of which I'm not against that. But if that was the case, then you should just let me do a random video whenever I want to, and then upload it on YouTube. But that's why I'm saying from next week onwards, I'm not uploading any session. Even now, if I don't want to, I won't. Because I told people that we're going to attend and then they don't. So you'll forgive me, guys, if it seems like I'm rude. And I always know that people will come with excuses of network and being sick and everything. But I'm not stingy with information I will give you. It's fine. So we have two types of endoplasmic reticulum. We have the rough endoplasmic reticulum, and then we have the smooth endoplasmic reticulum. This is what I want you to know, the difference between the two. The rough endoplasmic reticulum is abundant in cells that synthesize and secrete proteins. For example, your pancreas. Right? Your pancreas has to transport and it has to secrete digestive enzymes, right? So meaning that if you see a cell that has more of endoplasmic reticulum, it means that that cell synthesizes proteins, a lot of proteins, right? And then with your smooth endoplasmic reticulum, it doesn't play any role when it comes to protein synthesis. It has nothing to do with protein synthesis, your endoplasmic reticulum. But it does what? It does lipid metabolism, right? It's involved in lipid metabolism. Guys, if I'm too fast, please tell me. I'll slow down. If there's something you didn't understand, tell me. So if there can be a question to say which one of the endoplasmic reticulum is responsible for or is involved in protein synthesis, the answer to the question will be rough endoplasmic reticulum, because your smooth is not involved in protein synthesis. I repeat, your smooth endoplasmic reticulum is not involved in protein synthesis, but rather it's responsible for lipid metabolism. What do we mean, or what do we talk about when we say lipid metabolism? That is your fat and cholesterol synthesis. So it synthesizes, instead of synthesizing proteins, it synthesizes what? Fat and cholesterol. So meaning that the cholesterol that you see that is between the fatty acids of the plasma membrane, where is it from? It's from the smooth endoplasmic reticulum, right? So it synthesizes fat and cholesterol, and it breaks down fat and cholesterol, right? And it's also, your smooth endoplasmic reticulum is also responsible for detoxification of drugs, right? Let me. Hey guys, even that's an error. Yeah, detoxification, right? That is all you have to know about smooth endoplasmic reticulum. So you now understand the difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Now let's move to the next organelle, which is because these ones are related. Let me go to the Golgi apparatus. This one here. So your Golgi apparatus is responsible for modifying, packaging, and transporting the proteins. So this is simple, guys. Look at this. This is just a link. Detoxification of drugs. So look at this. You have your ribosomes on top of your endoplasmic reticulum. So what do your ribosomes do? They synthesize proteins. Where do those proteins go? They go into the rough endoplasmic reticulum, right? They go into the rough endoplasmic reticulum. And then from there, they will you will have some a vesicle. Remember, I told you about the vesicle when we were discussing the skin also. I don't know what we were talking about, but I told you about the vesicle. So there's something called a transport vesicle. It will, let me explain this. If this is your endoplasmic reticulum, right, and you have your proteins, they will sort of like pinch off or get into this vesicle, which I said it's like a bubble. Then that vesicle will go to the Golgi apparatus, just like you can see here. This is a vesicle. It's coming to the Golgi apparatus. And then when those proteins get to the Golgi apparatus, what will happen to those proteins? Your Golgi apparatus will do what? It will firstly, that's the first thing that you have to know about the Golgi apparatus. It will modify those proteins. After modifying the proteins, it will do what? It will package them. It will package the proteins. After packaging them, what it will do? It will transport them. So it does these three things. It modifies them, it packages them, and then it does what? It transports them using Golgi vesicles. So you will have another vesicle there after the protein has undergone modification and packaging. You will have another vesicle, a Golgi vesicle, as they show you. So that Golgi vesicle has the one has proteins, right? And then those proteins can move to three separate places. I repeat, the Golgi vesicle can go to three separate places. Firstly, it can transport those proteins to the outer part of the cell, which will be the extracellular fluid. For example, your mucus. Your mucus, after it's manufactured, it has to go out of the cell because it has no function inside of the cell, right? So that's number one. Number two, those proteins can go to the cell membrane itself because we just saw that cell membrane has a lot of proteins. So the proteins can be manufactured and then taken to the cell membrane itself. That's number two. Number three, the proteins can be manufactured and then taken into the cell. They are manufactured into the cell and then they are moved from one place to another in that particular cell, the very same cell, right? So for example, you can get your hydrolytic enzymes, right? Your hydrolytic enzymes, which will move from the Golgi apparatus to your lysosome. So your lysosome will be our next organelle that we'll discuss. But for now, what I want you to know about the Golgi apparatus is that it modifies, it packages, and it transports proteins. Then it can transport them to three different places, out of the cell, into the cell, and it can transport them to the plasma membrane. I hope we are clear. And then those ones that are going to the lysosome, what is the function of the lysosome? The lysosome is formed from, it's not like these proteins will go and diffuse into the lysosome. No, the lysosome is what? Is the Golgi vesicle itself. It parts from the Golgi apparatus. But for the mere fact that it didn't go out of the cell and it didn't go to the plasma membrane, it now becomes what? Lysosome. I hope I'm making sense and I'm not confusing you. So if they can ask you where does the lysosome come from, you tell them that it parts from the Golgi apparatus. And then it contains digestive enzymes. And just write DE. It contains digestive enzymes. And it's mostly found in white blood cells. Your lysosomes, if you can check the white blood cells, they have a lot of lysosomes. To be specific, which white blood cells? Your phagocytes. Because what do? They digest foreign particles. So that is the function of white blood cells. That is why white blood cells have a lot of lysosomes. But sometimes it happens that the cell digests itself. It self-destructs. For example, if if it has lack of oxygen, it can self-destruct. These hydrolytic enzymes, those digestive enzymes, I mean to say, they can digest the cell itself and the surrounding organelles because it lacks oxygen. And sometimes it's because it can be injured. Sometimes it's because of excessive, when it has too much vitamin A. That is all you have to know about lysosomes. What is important to you is that you have to know that it parts from the Golgi apparatus. And since mostly your lysosomes are confused with your peroxisomes, right? Your peroxisomes are, in terms of size, they are smaller compared to your lysosomes. But what you have to understand is that your peroxisomes come from the endoplasmic reticulum. So they part from the endoplasmic reticulum, whereas your lysosomes part from your Golgi apparatus. Guys, those are the differences that you have to know. You'll be asked such questions. I'll repeat, your lysosomes part from the Golgi apparatus, whereas your peroxisomes part from your endoplasmic reticulum. And your peroxisomes, the main function of peroxisomes is that they convert what we call free radicals, right? Do you know what is a radical? Do you know what is a radical? Okay. If the great example for this one, we all know that H2O is a water molecule. If I take out one of the hydrogens and then I'm left with something like this, right? We call this what? A hydrolytic peroxisome. Not peroxisome, radical. Right? So what a radical is it's an element that lacks an electron. Because you remember when you were back in high school, when you were drawing off Bow diagrams, that electrons must always be in pairs, right? So if it lacks an electron, it will go around the body looking for what? For that one left electron. And free radicals they can damage the structure of the proteins and nucleic acids. So that is why these peroxisomes have to take care of the free radicals. So when it comes into contact with free radical, it distracts it. Because if it doesn't, that free radical, because it is so eager to find another electron, it can end up destroying your proteins and your nucleic acids. So what does this peroxisome do? It converts the free radical to hydrogen peroxide. They'll also ask you this question. What does the peroxisome convert the free radicals to? It converts it to hydrogen peroxide. And if there's excess of this, it can further convert this hydrogen peroxide to water. I'll repeat. Your peroxisomes convert free radicals to hydrogen peroxide. They can further convert the hydrogen peroxide to water. That is all you have to know about the peroxisomes. And if they ask you what type of enzymes they contain, you think of peroxide. Peroxide, they contain oxidase enzymes. From the word peroxide. You take this oxide, you think of oxidase. So they contain oxidase enzymes. I hope you're clear. But the major difference I want you to know is that these peroxisomes, they part from the endoplasmic reticulum, whereas the lysosomes part from the Golgi apparatus. And then the next one, because you can see we have okay, let's go to centrosome. Your centrosome is just your centrioles, right? Your centrioles, what you have to know about your centrioles is that they generate microtubules, right? Your centrioles generate microtubules. And that they are direct formation of mitotic spindle during cell division. Like when you were doing, you know that you would have a centriole there, and then have another one, then you would have mitotic spindles that will hold the chromosomes and pull them apart. Yeah, you see. So what you have to know is that they generate microtubules, and that the direct formation of mitotic spindle during cell division. That is all you have to know about your centrioles. And then, since we spoke about microtubules are a form of cytoskeleton. This is not a good diagram because it only shows microtubules. Guys, this is messy. Let me just try to erase some of the things so that I can explain what I want to explain. Okay. So your microtubules, they are part of what we call cytoskeleton. So what is your cytoskeleton? Your cytoskeleton is the muscles and bones of your cell, in simplest terms. Your cytoskeleton is your muscles and bones of the cell. And then we have three cytoskeletons, the filaments that you have to, that constitute of the cytoskeleton, that make up the cytoskeleton. Number one, it's your microfilaments. Sorry. Number two, it's your intermediate filaments. I'll just write interne because I've already written filaments there. So you have your intermediate. Don't write inter filaments. Let me write it carefully. Hey guys, sorry for the handwriting. Intermediate filaments. And then number 3, you have your microtubules. So your cytoskeleton is made by microtubules, intermediate filaments, and microfilaments, right? So what you have to know is that your cytoskeleton basically is just a network of protein structures, because these are protein structures. But as I said, in simplest terms, it's just the cell's muscle and skeleton. And what you have to know is that your microfilaments are responsible for cell motility and producing changes in shape. So when we speak about cell motility, cell movement, right? That is where your microfilaments come into play. I'll just write cell motility. And then you also have to know the 2 types of microfilaments, because we discuss them in detail when we do the muscular system or muscle physiology, right? So the 2 microfilaments that you have to know, it's your actin and your myosin. And your actin is the thin filament, whereas your myosin is the thick filament. But they won't ask you that for now, but they will ask you in your exam. Obviously, by that time, you would be done with your muscle physiology. So if you get to muscle physiology and you didn't understand cells and tissues, and you don't even know that this is part of the cytoskeleton, it will be difficult for you to understand muscle physiology. Cell mobility, cell motility, one and the same thing. But cell motility, I think I'm wrong. It's cell mobility, guys. But is there such a word of? It seems like my brain is playing games with me. Let's say cell mobility. I'll check it. Or do we have the word motility? Cell motility. Guys, go with mobility. I don't know what's happening with my brain, but I'll check. Mortality is death. But I didn't write that mortality of MORT. I'll check. Man, it's cell motility. But I think cell motility and mobility is one and the same thing. So my worry was just that, is there such a word as motility? Because sometimes, guys, my brain plays games with me, because I try to simplify things to a point whereby I even say words that I don't think exist. Like earlier on the other session, I said what I said, did I say productivity? Like, Jesus, sometimes I just feel like the English bundles just get depleted out of the blue. So yeah, it's cell mobility or cell motility. But if you think motility is correct, yes, how? I know my bundles are still a. You see, I buy bundles, I buy data English bundles every hour. So it can move me here and there, but I'm stable. So going to your intermediate filaments, they help form desmosomes. Remember that when we were doing desmosomes, what did I say? I said in between we have cadherents. Oh, guys, we're left with 10 minutes, but I think we're left with one more organelle to discuss, and then we'll be done. Then I said we have plaques, right? And then I said from each plaque there are filaments. So what are the name of those? What is the name of those filaments? It's what? Intermediate filaments. And then your microtubules are made up of the protein tubulin. Microtubules are made of protein tubulin. And they determine the shape of the cell. So whether the cell is circular or whatever, that is determined by microtubules. Then the last thing that I want us to focus on is your cell extensions. So cell extensions, this is it even here? It's not. We covered everything. You see, there's no organelle that I missed. We spoke about plasma membrane. I told you what is a chromatine. Yeah, guys, we spoke about everything. There's nothing that I missed here. The one of vacuole is not really that applicable in animal cell, especially as humans. So forget about it. So I think I told you about each and every organelle. Again, I'm telling you, it's very easy to forget this stuff. As easy as it is, and since you've been doing it from high school, trust me, it's very easy to forget this stuff. So please repeat, know the functions, know the structure, because that is where you get easy marks. And then, yeah, since I think I covered everything, let's go to cell extensions, because they are not even shown on this diagram. And then I think that will be it. I can wrap it in the remaining 7 minutes. So with your, let me rather type instead. So your cell extensions, we have 3 of them. We have your cilia, your flagella, and microvilli. We have your cilia, flagella. Guys, I won't write everything in full because time is against me. And microvilli, you know how to write microvilli. M-I-C-O-R, you know, guys. So your cilia, it's involved in, it moves the particles, or it moves molecules that are on top of the cell. Like, for example, let's say this is your cell, and then it has cilia, right? So your cilia would, for example, move mucus. It would move the mucus. You see, just like your fallopian tube, it would move the ovary. See, the oocyte, it would move that oocyte. So your cilia, what you have to know about it is that it just moves the particles or the molecules that are on top of that particular cell, right? A great example where you'd find your cilia is in like your respiratory system, because when you breathe foreign particles sometimes. So there's a lot of mucus, and it gets trapped in you. The foreign particles get trapped in your mucus, so the cilia will have to move the mucus, right? And the only cell in the human body, I repeat, the only cell in the human body that has flagella, because flagella is, if this is your cell, flagella is seen in bacteria, but the only cell in the human body that has flagella, because flagella moves the cell itself. whereas cilia doesn't move the cell, but it moves the material on top of the cell. Do you get the difference? Whereas flagella moves the cell itself. So the only, as you can see by the drawing, the only human cell that has flagella is the sperm cell. That's the only one. And then going to the microvilli, you see, people tend to confuse microvilli with cilia. Microvilli, if this is your cell, because it's finger-like projections, right? It's finger-like projections, it's something like that. So your microvilli does what? It increases surface area firstly, and it does not involve microtubules. What do I mean by that? These two, your cilia and flagella, they are made by microtubules. The reason why cilia is shorter than the flagella is because the microtubules didn't extend that much compared to here. So this one, the microvilli doesn't involve microtubules. It's formed by a core of actin filaments. It's just a core of actin filaments. And what do I mean by microvilli increasing surface area? Because let's say, because the great example of this one would be your intestines. There's a lot of microvilli in your intestines, or yeah, for absorption, right? So let's say this was your intestine. Let's say this was the length of intestine. So it would absorb, let me draw, yeah. Let's say this were food molecules. So it's 1, 2, 3, 4, 5. It will only absorb 5. For the same distance, now that the intestines have microvilli, they can absorb more than that. Because for the same distance, let's say that is your intestine, it is this microvilli. It is this microvilli. See, it's same distance as this one, same length, but it can now absorb here. The particles will stick here, stick there, stick there, stick. So it will obviously absorb more than 5, just like this one. So that is what I mean by increasing surface area. So that's it, guys. Though that was all of the organs that you need to know, organelles, what I mean to see. And yeah, I think that's just about it. We are left with cell physiology and cell division, and then we can move straight to the tissues. But I don't think we'll have a class later today. It will be unfair to you guys. You have to rest, like, seriously. So with the minute that is left, what is it that you want to see? Didn't you understand something? Wanting to clarify something? Guys, talk to me. We have a minute left. Do you have a question? So it will kick you out. Don't bother coming back with time. Yeah, I hope you'll enjoy your biology session. Yeah, I wish you all the best. Have a great day, guys. I love you, but God loves you more. Bye bye.